So with the plant disease monitoring for scheduling control measures and also predicting crop yield losses. And let me repeat it, why we should be interested in monitoring these diseases or insect pests. We are trying to minimize unnecessary pesticide sprays so that we can have value by the end of the cropping season. Now with disease monitoring, it is critical to know the amount of disease that is present in crops. Presence or absence of the disease may not be critical. The disease has to exceed a certain threshold before it reduces crop yield. Even if the disease is there, is it present at such a level which justifies spray of fungicides? That is the question. For example, small amount of disease could have little effect. So should we spray the fungicide? The amount of disease is measured as the proportion of crop population that is infected. Actually, this is what we call disease incidence. If we are looking at the population of plants in a field and we try to estimate the number of plants which have been infected, it could be 50%, or less than 50%, 60, 30%, and so on. That is actually pointing at disease incidence. But if we are looking at a specific plant, an individual plant, and we are looking at the proportion of the area of a plant or a plant organ with the disease, it could be the leaf which has been affected. That one is looking at disease severity. We could have one plant with a high disease severity, yet the incidence of the disease is low in that field. So these are some of the things which we have to look at when assessing plant diseases. Assessment of plant diseases and the effect on crop yield. Now, developing descriptive growth stage key for a particular crop species in question, developing methods to assess the incidence and severity of disease, and we should relate it to the growth stages. We should also develop statistically sound method of sampling crop populations for the assessment of the amount of disease. Also estimating the negative impact of a particular level of disease on crop yield and the quality. Overall, we should be able to evaluate the economic benefits from various methods available to reduce the amount of disease. We can assess the crop growth and development. There are descriptive and pictorial growth stages, growth keys. For example, in cereals, we can have those growth stages. So we should be able to say at this stage, is this plant affected? How much is it? To what extent is it affected? So we relate to growth stages. I mean, if at the end of the growth period, at the maturity, we often see a lot of diseases coming towards that end. But if you look at the crop period, it could be okay. Probably there's no need to do sprays at that end there. So methods of assessing disease incidence and surveying provide objective measurement for comparison. They could be simple and quick to use. They should be related to an identifiable growth stage of the crop, provide adequate sample for the area. Measurement of disease incidence and severity. Percentage of infected plants usually points to disease incidence. So disease incidence is based on counting infected and uninfected plants. Estimating photosynthetic area of the crop that is diseased, a proportion of leaf area affected. Probably we are now looking at severity of the disease. So disease severity assessment relies on visual judgment. This varies from person to person. We can use disease assessment keys, use published keys which were developed by others. For example, we have got this old key, but could be very useful. You have got severity. You are looking at an individual plant where we have got a scale from 0.1 up to 100. And we have got those descriptions for each category. Depending on what you are seeing, you put a percentage or you record a percentage. Then we also have this other one which was developed by the British Mycological Society for Phytophthora investans. Now, if you look at the individual leaf with those spots, they can be scored so that we come up with a number and say this is 2%, 5%, or even 50%, depending on how we are seeing because we'll be comparing those spots to the actual leaves. and give percentages. Some of the assessment tools may involve use of remote sensing techniques. For example, Puccinia graminis, we can use spore traps, just like what we do with the insects. You can trap the spores and determine the quality of spores. This can give you an idea of the indication of the presence of the disease. There are also what they call electronic scanners, probably, which can detect these spores. Or infrared aerial photographs. These can actually pinpoint diseased areas on the plant leaves or on the plants. Or use of video cameras and image analysis. I have actually seen some of these methods being used at Sheffield University by people who are doing plant pathology. So, sampling for disease incidence and severity assessments. So, samples of crop units, leaves, flowers or inflorescence, etc. can be taken at random from a field. Walking across a diagonal or a W or a diamond pattern across the field and taking a sample every 10 paces. Alternatively, standard quadrats can be placed at random. Sampling intensity and standard error. In fact, sampling intensity is very important because they affect what we call the standard error. This is now a field of statistics. The more samples you take, you can actually reduce the standard error or a lot of variation. Now, you can check your sampling intensity and calculate these standard errors. Where you have that arrow, probably that is the ideal sampling technique. But from experience, other scientists will have a number of samples which they think is necessary to come up with a realistic prediction of the disease incidence. Probably after doing this thing over and over again, people will develop protocols for doing this kind of thing. Assessment of crop losses due to disease. So, effect of different levels of a disease on crop yield. How do we do it? We can use the experimental approach, setting up experiments in which the level of the disease is controlled, using different levels of inoculation or treatments with the fungicides, nematicides, and the bactericides. We can put a fungicide in some plots, and some plots we don't put fungicides. Then we compare. The relationship between disease progress and yields in plots can also be established. The assumption is that the biocide has no effects on yields in the absence of the disease. In other words, if we use the fungicide, we are saying the fungicide is not phytotoxic to the crops. And usually most of these recommended fungicides may not be that toxic to the crop. And indeed, the method of using fungicides has been successfully used to assess the impact of diseases on crops. A comparison of epidemic progress and yields can be obtained from experiments using susceptible and resistant cultivars in the same plant species. Regression analysis is commonly used. This is a statistical approach which gives us the relationships of presence of the disease and the crop yield loss. So we have what are known as critical point models. This is actually regression. The relationship between the amount of disease and the percentage loss in the yield. We can develop linear regression equations if we have found them to be statistically significant. So we can plot disease intensity, which is your x-axis, versus yield loss, which is your y-axis. And sometimes we get a relationship. For example, like the one for powder mildew, which was y equals to 2.5 square root x, where x is the percentage of mildew. So this was actually done by others. The relationship may differ depending on the disease. For example, with rice blast in Japan, they found that the relationship was y equals to 0.5x. This was regression. X is the percentage of blasted parts. So it may differ. But then the model will be applying to the crop concerned. These are some of the graphs on severity of disease at time, and they show the critical point. What I will do, if you are interested in them and you have the time, I will supply you with the reference where I got this graph so that you can read about it. Multiple point model, this is simply a regression analysis, a multiple regression analysis which considers a lot of factors in predicting the loss due to the diseases. So a lot of factors are involved, and you end up with that sort of equation, as long as you know that sometimes we involve a lot of factors to predict crop yield loss due to diseases. It could be the number of spores present, temperature, rainfall, soil type, etc. Whatever factors which we think could be useful in predicting the yield loss. So there may not be any need to sort of memorize this multiple regression model. Again, we also have these severity of disease graphs, which are showing the progression of severity with the time. So we can actually do a quantitative analysis where we relate the disease intensity and yield. Now, in most cases, if you relate it to disease intensity to crop yield directly, you get a straight line pointing to the loss in yield. If you now plot the yield loss, you get an increase in yield loss as you increase disease intensity. This is actually expected. Here we've got several graphs. Probably it could be different varieties being compared in terms of their susceptibility to the disease, or different control methods to find their efficiency in controlling the disease. What is important is to plot disease severity against yield or any other plant measurement. Another interesting issue concerning diseases, if we monitor them under field conditions, we may also want to predict outbreaks so that we are better prepared for the control measures. So we have a disease forecasting that is predicting of disease occurrence. It is important in regulating chemical sprays or biological control agents. If we know when the disease is coming, we can prepare for it. And sometimes there may not be any need to spray if you are sure that the disease is not coming. So knowledge of the biology of the disease and the prediction of outbreaks is very important. Planting virus-infected potatoes will cause the crop to be infected. But this is not forecasting. What is plant disease forecasting? It is combining weather data and the biological data to predict the disease incidence. So you need your environmental conditions to consider, especially weather, because the spores are affected probably by humidity and so on. So we can use this disease monitoring as an extension for forecasting the presence of the disease. Now, which are the diseases which are suitable to do forecasting? The disease should be economically important. The disease should be above the threshold level to cause loss. If the incidence of the disease is very low, probably there's no need to do the forecasting. If the disease rarely reaches the threshold, it may not be worth forecasting. Example, blister blight fungus in tea can occur at 35% or more. This can cause a significant economic damage. This can be the basis of probably engaging control measures. So disease control measures, routine control measures, are expensive and wasteful. Seed treatment can be important. You can reduce the occurrence of the disease. Use of resistant varieties can also help to reduce sprays. So fungicides sprays need prediction of diseases. Efficient control measures are available. It's pointless to do disease forecasting if there are no control measures. If you just say, okay, the disease is now arriving, and you don't take any control measures, then that information is not used. So what do we hope to achieve by disease forecasting? We want to reduce the incidence and reduce cost of disease control, because we will be better prepared. If we know that the disease is coming, then we can prepare for it. If we know that the disease is not coming, then there's no need to go about spraying fungicides. So we can only spray fungicides when it is really necessary, or we can use protected measures. This is just an interaction of the pathogen, conducive environment, and susceptible host that will produce a disease. You need the pathogen, you need the conducive environment, and you need a susceptible host. Now, just briefly looking at the methods of disease forecasting, there's correlation between disease incidence and weather. So if we know some information about weather, we can predict, and we can use now computer modeling to facilitate this prediction. Factors which are important are rainfall, temperature, humidity, wind, light, sunshine, and cloud cover. On sprangial for development, spore development, duration of spore production, number of spores produced, dissemination of spores, spore germination and infection. All of this data, if we have it, we can precisely predict whether the disease is going to occur or not. Empirical correlations using several factors in multiple regression, so that we can actually predict the occurrence of the disease. monitoring inoculum, not effective for potato red blight and grape down mildew. It could be suitable for rice panicle blast caused by Pricularia oryzae, predicted by the number of spikelets infected throughout inflorescence development. Trapping iridospores of wheat rust on Vaseline coated slide or glass rods. We can also use this to predict whether the disease is likely to occur. Monitoring spore concentration in the atmosphere, we can check whether there are spores in the atmosphere. If a certain threshold is reached, we get prepared for sprays. A spore sampler with the sticky sides is actually used. Sampling for sclerotia in soils, sclerotinia, sclerotia can actually be sampled. Nematode sampling can also be done. Now, microscopic propagules. Soil samples are sequentially diluted with soil or water to give dilution series that is placed on a medium. Various antibiotics, fungicides, and dyes are added to the medium to suppress growth of saprophytic bacteria and fungi. I think this method is commonly used to assess the presence of the disease. Also, trap or test plots. We can grow susceptible cultivars. If they are infected, they can give us an early warning of the arrival of the disease. Then we will start spraying the other crops. We just plant a few plots which we will keep on monitoring. If they are infected, we start spraying other crops. These are just the leaves showing infection. Probably using photographic images, probably this is what you get. Those red areas indicate infection better than that other leaf. So this is it for plant pathogens or diseases.